Hello, how are you guys today? So we're going to make a quick video about egg osmosis. This is going to supplement our lab number 6, which is movement through the membrane. So this is going to be a nice demonstration on how osmosis works. So the first thing you should do on the worksheet is follow the instructions to fill out the circles and red blood cells. So you're going to watch a video on YouTube to do that. So please do that. The next thing that we're going to do is this part down here, which has to do with egg tonicity. So let's greatly magnify what's taking place in a red blood cell by using eggs in class. So I don't have very much time to make the video, so we're not going to see a huge weight loss or weight gain with the eggs, but that's okay. I think you'll still get the idea. So what you're going to do is we're going to take the mass at the start of class, mass at the end of class, and then you're going to fill out the rest of the worksheet. Did the egg gain water, lose water, or stay the same? The next thing you're going to do is you are going to answer the questions and state whether the eggs are hypertonic, isotonic, or hypotonic, comparing it with the solution that's found on the outside of the egg. So what we're going to do in each of the beakers is we're going to put a different solution. In the first, with egg number 1, we're going to put the egg in distilled water. The second egg is going to go in a ringer solution. And the third egg is going to go into a solution that's going to mimic a 10% sodium chloride solution. We're going to put it in K-Row syrup. In the picture, you're going to draw an arrow and tell me whether the egg lost fluid, whether the egg gained fluid, or whether the egg stayed the same, okay? And you're going to do that for each of the eggs, eggs 1, 2, and 3. Now, at the beginning of today, I took some eggs and I put them in vinegar. When I did that, it caused the eggs to lose their outer shell. So the outer shell is gone. This is not a hard-boiled egg. It's a regular egg that I took out of the refrigerator and then put in vinegar. And I did that so that these eggs could mimic a cell. So around the egg is a cell membrane, and it's nice and soft. They're all about the same, right? And what we're going to do is determine how fluids that are found around a red blood cell cause red blood cells to either gain water, lose water, or stay the same. So first of all, what we're going to do is we're going to weigh egg number 1. Egg number 1 weighs 67.5 grams. I'll go ahead and record that. I also want you to record that in your worksheet under egg number 1, mass at the beginning of class. So we'll go ahead and take the egg and we're going to put it into the beaker that is labeled egg number 1 distilled water. Okay, then what we're going to do is we're going to measure and see, did that egg gain water, lose water, or stay the same? Okay, so now we have egg number 2. Egg number 2, we're going to put in a solution that's called ringer solution. Now this particular ringer solution is amphibious or amphibian ringer solution. So what we're going to do is see if this has the same tonicity as our egg. So we'll see if it gains, loses, or stays the same weight. And by that, we'll be able to tell if this amphibian ringer solution is the same tonicity as the egg or whether it's a different tonicity. So I guess we'll have to wait and see. I was kind of hoping it was the same. If it's the same tonicity, it would be called isotonic. If it's isotonic, it should not gain or lose weight. So we'll see. Let's see what happens. This will be our unknown, okay? So we'll take our egg number 2. We'll go ahead and weigh that egg, and it's 70.5 grams. I'm going to pause the video and record it on my egg number 2 worksheet here, and you guys can do the same. Go ahead and record that under egg number 2. Alrighty, I recorded that under egg number 2. Now I'm going to put the egg into beaker number 2, which is a Ringer's solution for amphibians. Interesting, we'll have to see what happens. Okay, so now we have our last egg, egg number 3. It's right here. Now this egg is going to go into a solution that's going to mimic a 10% salt solution in a human body. So instead of a 10% salt solution, which didn't work very well, I am using Karo syrup, which works really well. So let's go ahead and weigh our egg to get our starting weight at the beginning of class. This egg weighs 62.4 grams. I'll go ahead and pause the video and record this weight while you do the same. Alrighty, I've recorded the weight. Now I'll go ahead and put my egg into the Karo syrup. There we go. I am going to go ahead and pause the video and go ahead and cover the egg with Karo syrup, and then we'll come back after some time and we'll weigh the eggs again and see what happens.